So this lecture is going to be covering carbohydrate catabolism, which is in chapter 5 of your textbook. So carbohydrate catabolism is also known as cellular respiration, and there's 3 main parts, and we'll talk about all of those as well as their location and where they happen in the cell. All right, so let's get started. The first step of carbohydrate catabolism is called glycolysis. So I'm going to write carbohydrate catabolism, and then I'll put the first step, glycolysis, up there. So glycolysis is shared not only by carbohydrate catabolism, but fermentation also includes glycolysis. So in glycolysis, you have the full, or in carbohydrate catabolism, you'll have the full breakdown of glucose. In fermentation, glucose won't be broken down completely, and you won't get as much energy from it, not nearly as much energy as carbohydrate catabolism. But nonetheless, they both have glycolysis as part of the process. Okay, so we're not going to talk about each part and all of the different reactions that go on with each one of these steps, but we are going to focus on the main inputs and outputs of the first steps, and then we'll talk in detail about the electron transport chain. So with glycolysis, the input is a molecule of glucose. So we put in a molecule of glucose, and just like the title implies, catabolism means the breakdown. So glucose will be broken down. Glucose is a 6-carbon molecule, and after glycolysis, you will have 2 3-carbon molecules. You'll have 2 pyruvic acid molecules. So like I said, pyruvic acid is a 3-carbon molecule. Okay, so as we go through each of these steps, we're going to keep a running total of the amount of energy produced in each step, and we also need to keep track of what are called electron carrier molecules, and I'll explain more about that when we get to it. But glycolysis will yield 2 molecules of ATP. So technically glycolysis yields 4 ATP, but 2 are used in the process, so there's only 2 net ATP produced after glycolysis. Okay, so the other thing we have going on here in glycolysis is when we take a step back. So we're going to start talking about what are called electron carrier molecules. And essentially there's going to be molecules that their function for this is to be reduced, meaning to accept an electron and carry it through and then be oxidized and dump their electron out in the electron transport chain. So remember, reduced is to gain an electron, oxidized is to give away an electron. And sometimes I make an O and push it away for oxidized and reduced. And for some reason that helps me remember, so perhaps it will help you. All right, the name of the electron carrier molecule that we'll talk about first is called NAD. So NAD will be reduced in glycolysis to and accept an electron. So when you see the H at the end, that means that it has gained an electron. And you can, I don't teach chemistry, but you can ask your chemistry instructor on the ins and outs of that. But when there's an H added, you get the electron added with the hydrogen molecule. So NAD got reduced and became NADH. Okay, And it's going to just hang out with its electron, and at the very end, it's going to dump it out in the electron transport chain. Okay, so let's move on. So with these two pyruvic acid molecules, these will be converted into two molecules of acetyl-CoA. And so the acetyl-CoA is what's needed as the input for the citric acid cycle, which is the third step. So the pyruvic acid can't go directly in there. It must be converted to the acetyl-CoA to go to the citric acid cycle. This is called the prep step. And the prep step is the second step in carbohydrate catabolism. During the prep step, we get two additional molecules of NAD that get reduced to become NADH. No ATP is made during this step. There is some carbon dioxide that's made. At this point, I'm going to stop and we're going to get a little running chart going before we get into the citric acid cycle and into the electron transport chain. So this chart I've made, it's kind of a glare. This is NADH at the top. This is FADH2. And then ATP. And then I just put a G for glycolysis. Whoop, there we go. G for glycolysis, P for prep step, C for citric acid cycle, and E for electron transport chain. It's important that you know kind of these running totals as we go. So for glycolysis, we have two molecules of NADH that were produced. FADH2 is another electron carrier molecule that hasn't been introduced yet, so no FADH2 yet. And then we had two molecules of ATP produced. All right, with the prep step, we had two NAD produced. We have no NADPH and no ATP. Now we're ready to move on. So going back to our process here, we have our two acetyl-CoA that are now ready to go into the citric acid cycle. Okay, so the citric acid cycle is also known as the Krebs cycle, if you've heard that term for it. In the citric acid cycle, we have the input of the acetyl-CoA, and then we have quite a few outputs. We have two ATP that are produced. We have some carbon dioxide. And then we get some more electron carrier molecules. We get 6 NADH that are produced. And we get 2 of the FADH2 molecules. And again, those are electron carrier molecules. It starts as a molecule of FAD, and then it's reduced to become FADH2. All right, so let's fill in our chart. In the citric acid cycle, we had 6 NADH, 2 FADH2, and 2 more ATP. All right, so our last step is called the electron transport chain. And this is where all these electron carrier molecules are going to be oxidized and give off their electrons, and that will be used for something. So let's jump into that. First, let's talk about the structure of the electron transport chain. The electron transport chain is a series of enzymes or proteins that are embedded in a cell membrane. All right, and depending on the type of cell, these will be embedded in a different membrane, and we'll talk about the locations of those when we talk about that after we get through all of that. But again, these are a series of enzymes that are embedded in a membrane. And depending on the organism and the genus and species, these enzymes will be different. So the organism can kind of mix and match what enzymes are present there, and those can actually be characteristic or be ways that we can identify a certain organism or identify a bacteria. So the presence of a certain enzyme might be characteristic of a certain type of bacteria or a certain organism. For instance, the oxidase test that you perform in the laboratory, that's looking for the presence of a specific enzyme, cytochrome C oxidase, and if that's there, it can give you a clue to what type of bacteria you might be dealing with. Okay, so we have a series of enzymes. Now, what is next? So these electron carrier molecules are going to give their electrons to the enzymes. If it's a molecule of NADH, it will give the electron at the beginning of the electron transport chain, and it will be able to produce the most ATP that way. The FADH2 molecules will provide their electron in the middle of the electron transport chain, so they won't be able to make as much ATP as NADH. Okay, so the electron carrier molecules will be oxidized. They'll give their electrons away, and the enzyme, there will be molecules in the enzyme that will get reduced because they'll accept the electron. But it doesn't keep this electron. So it just got oxidized. It's going to immediately give that electron away and get oxidized again. So then the next enzyme gets reduced by accepting it, and then it's going to give it the electron away again and become oxidized. So this is called a series of redox reactions because they're being reduced, oxidized, and so redox. It's a series of redox reactions. That's going on with these enzymes. All right, so at the end here, this last electron, or I should say the last enzyme, it needs to be able to get oxidized and give that electron away in order to receive the incoming electron, the next one that's coming down the line. So it needs what's called a final electron acceptance.